All right, hi everyone. My name is Matt Fainoff. I am the Chief Technology Officer and President at Biosurfaces Incorporated. I'll be serving as the moderator for today's webcast that focuses on perianal fistulas. Many of you may not have heard of this affliction. I know, truth be told, when I first started, I did not know what a perianal fistula was. So in layman's terms, and we'll get into this more as we talk to our panelists, really a perianal fistula is a small tunnel that forms adjacent around the anorectal area and typically extends out into the skin. Although most are spontaneous, these can happen with increased frequency in certain gastrointestinal conditions such as Crohn's disease. We've assembled a group of experts in different areas with the goal of providing you an overall picture of how this complication affects people, what is currently being done to treat these fistulas, issues with current fistulas, how Takeda is approaching the problem, what Biosurfaces is working on with Takeda to improve fistula treatment, and what the future may hold. I would like to introduce our panelists for this discussion. First, Dr. Vitaly Poilin, a board-certified colon and rectal surgeon, is an assistant professor in the Feinberg School of Medicine at Northwestern Medicine. Vitaly began his medical career as a nurse in the Ukraine, which I learned looking at the website at Northwestern, which I thought was very intriguing. From which he then transitioned into surgery. His main areas of expertise are in diagnosing and treating diseases of the lower and digestive tract. Dr. Vincent Ling, a biologist by training, is a senior director in drug delivery technologies, search and evaluation for the Center of External Innovation at Takeda Pharmaceuticals. Vincent is a biotechnology innovator with diverse background in protein engineering, cell line production, molecular immunology, stem cell differentiation, antibody, alternate scaffold technologies, and cell-based therapies. And I want you to say that 20 times fast and see how that goes. Mr. Nate Long is a biotechnologist by training and is a senior research associate here at Biosurfaces. Nate has been extensively involved with developing an electrospun perianal fistula plug for our company, which we call the BioSpun J plug with our partners at Takeda. Over the past 3 years, he has worked on developing the prototype, conducting benchtop studies, and assisting in preclinical assessment. And last but certainly not least, Dr. Mauricio Contreras is a plastic surgeon by training and is an instructor of surgery in the Division of Vascular Surgery Research at the Beth Israel Deaconess Medical Center. Mauricio's primary focus over the past 31 years, and I've been happy to know Mauricio for the last 30 years of those 31 years, is the research centered on preclinical assessment of new medical devices. He has been instrumental in developing the preclinical model used to examine the BioSpun J plug, implanting the device, and performing histological assessment of the explanted devices. So I'm really excited to begin this discussion. So I'm going to dive right in and ask the first question to Vitaly. Now, Vitaly, can you tell the viewers a little bit about the background about what perianal fistulas are and why they form?
Sure. So as you started to explain, Matt, the fistulas are little tunnels that go from inside of an anus and usually come out to the outside. We have little glands on the inside. We actually don't know why we have them. We know why skunks have them, but we don't know why we are in humans. And mostly spontaneously, some people can develop infections. Half the time when this infection develops, it's a drain and goes away. But half the time, that little opening on the inside never completely heals. And there is stool and bacteria that keeps on getting inside that little tract, essentially creating this low-grade chronic infection. And then people will present with complaints of swelling, drainage, discomfort. Oftentimes it's just annoying, uncomfortable, but it can cause a pretty significant infections requiring urgent visits to the hospital, trips to an operating room, and such. So for most part, as Matt said, these are just bad luck. We don't know exactly why they form, and we don't know exactly why in some people they don't go away. There are some folks who have, for example, Crohn's disease, hidradenitis, a couple of other conditions that is related to the underlying problem with the tissue, and it essentially creates breaks in the mucosa and breaks in the integrity of the bowel, and then they can develop multiple and pretty significant issues with this multiple tracks that can be quite extensive and very disturbing. So that's kind of in a nutshell.
That's a great explanation. And how many people, are typically afflicted with these type of fistulas?
So
What do you see in your clinic and in sort of ballpark on a global sense? How many
yeah,
people are afflicted with these?
these are actually pretty common. It's difficult to estimate the true rates of these. Part of it is there are definitely folks who misdiagnosed, or they think it's something else and they don't necessarily present. Um, so the general estimates are we're talking about 2-3 people per 10,000 in people who don't have inflammatory bowel disease, and probably 7 to 8 people, 100,000 for folks who have something like Crohn's disease. So they're not uncommon. And as far as inflammatory bowel disease, so for Crohn's disease, this is the second most common single location of a disease, actually. So
Wow.
in that, it's pretty common.
Wow. And in terms of, you've shared some, stories of different patients, but what you've had to deal with in terms of, what these patients are, how they're afflicted. How does it affect their quality of life? Is what you see in the surgeon with your patients?
Yeah. So it's really a range anywhere from being an annoyance to people come in just like, I constantly have some staining in my underwear, or every week, every month, I go through these cycles of swelling, pain, discomfort that I have to deal with, and then it kind of starts all over again, to folks who have significant pains, have difficulty sitting, or actually have a much bigger abscesses that require drainage, antibiotics, difficulty with bowel movements, and it can be quite significant.
Wow. And can you talk a little bit about what you would do as a surgeon to treat these patients who have these fistulas?
So I usually will tell folks that this is the simplest complicated problem they can have, because we actually know exactly what to do about it, especially for non-inflammatory bowel disease related to called the cryptoglandular fistulas. If we cut them open and we literally lay them open, they will heal from inside out, and they have a great chance of success. It's about 93-94%. Once they recover from, it's a quick outpatient procedure. I did one today. It took me about 5 minutes in the operating room, and they're done. And this is luckily a vast majority of this fistulas. However, this is also the area where the sphincter muscle is, and this is what controls the continence. So if a fistula is deeper, or we're too aggressive and we open it up and too much sphincter muscle is cut, then we can heal the fistula, but people have trouble with control, have trouble with incontinence, and this is obviously not what anything kind of wants and likes. So then it's a sizable minority. It's in the cryptoglandular fistulas, we're probably talking about 20-30%, depending on what study you want to look at. We have to go a more complicated route. Usually for these fistulas, we start out with putting a rubber band. It's literally a rubber band hanging off the backside. It's almost like a piercing down there, but allows the whole area to settle down, inflammation to decrease. And then we go with other procedures that are designed to try to fix the fistula without affecting control. Procedures such as indirectly advancement flaps, when we take some normal tissue and kind of pull it over the opening, trying to cover it up and close the inflow, to what's called the lift procedure, ligation of intersphincteric fistula tract. When we go between the sphincter muscle, we find the tract, we cut it, tie it off. The success rate of these procedures is about 60-65% at the best case scenario. They're more involved. There's a longer recovery with much more limitations for what people can do. And obviously 60-65% is not great success rate. There is one fistula plug available on the market at this point, and essentially the idea that you put a with biodegradable material inside the tract, and your body kind of grows into it. These are easier technically to perform, but success rate currently is quite low. It's less than 30%, and so most of us don't do it for these reasons. There's also other variation of a plug, like a fibrin glue, which has a pretty similar success rate. So these are the things that are currently available. There are a couple of other things that are on the horizon, but they're mostly in the trials and not available to most patients who come in through the door.
Do you have, I mean, it's just sort of off, a little bit off of what we were thinking about in terms of a question, but, do you have any idea or thoughts in terms of the current fistula plugs? Why are they not successful? You think it would be biodegradable and it would grow in and things would happen. What is your thoughts in terms of what have you seen clinically that lead you to believe that there's a reason that they're 30%, effective or 70% ineffective?
Yeah. So, nobody knows for sure. I think one issue definitely is that it's end up being a high pressure area from both when people go to a bathroom and then they push and they, kind of dislodge those plugs and push them out, to the fact that probably they probably not approximating too well to the size of a fistula itself, creating kind of empty spaces probably in between, and then perhaps in addition that the tissue does not grow in fast enough. So essentially they kind of end up allowing some bacteria through because it's impossible to sterilize this area anyway. And then as it happens, it essentially, once it starts getting infected, it kind of floats away. Those would be my guesses for why.
That's great, great background and thoughts on that. So I think, why don't we, I think you've given a great background on this. And so let's bring Vincent into the conversation. So Vincent, Takeda's had a long-standing history for providing pharmacological interventions for various diseases, from gastrointestinal, oncology, and so forth. It amazes me that Takeda has been providing these services since 1781. So being able to deliver or provide a drug to a patient, for so many years is amazing in terms of Takeda's history. Can you just tell me a little bit about, and tell our viewers a little bit about Takeda's current efforts to provide, specific treatments, not necessarily for the plug or for the tract, but for gastrointestinal disease in general, such as Crohn's disease?
Right, yes. So Takeda has 4 therapeutic areas, and one of them is gastroenterology, or GI diseases. And within that, there's 3 segments that are of special focus. One is liver disease, another is motility, and the third is inflammatory bowel disease. And I guess Takeda recently made their mark in the pharmaceutical world by releasing a new drug called Entyvio, which is an antibody drug that is very, very effective in ulcerative colitis and Crohn's disease. And it's become a very good drug for treating these diseases because of minimal side effects. And it's really driving the entire franchise forward into possibly treating other inflammatory bowel diseases as well, too. And so that's where the drive has come from.
Oh, that's excellent. So, when I introduced you in our opening, I mentioned that myself, as well as many others, consider you to be an innovator in various spaces, having the pleasure of being able to work with you over the past 5 years. What led you to think about using alternatives or complementary technologies to help treat perianal fistulas? Being from the pharma company, you were looking at not just the pharmacological intervention, but other ways to treat it as well.
Yeah, so my background is that I've been in biotech and pharma for the past 27, 28 years. And for the past previous to my 7 years at Takeda, I had been in biologics discovery, so protein engineering, molecular evolution, things like that, trying to create new drugs. And 7 years ago, when I joined Takeda, I realized that there's another world out there other than inventing new drugs that can enable therapies. And so I proposed when I joined that I would explore drug delivery technologies. And the thing that attracted me most to the idea of using electrospun materials was the concept that here's a material that can cause a physiological response in the absence of a drug, right? And so that's really important because now you have a material that has a drug-like property by having cells respond and infiltrating it and causing almost like a regeneration of tissue on site, right? But you're not adding any growth factor or any drug that elutes out to cause this to happen, just using the material by itself. And so to me, that was a new modality. And it's in that gray zone of biomaterials that have biological activities that I found extremely interesting, because that's generally not an area that people look for in terms of a drug therapy.
That's great. And can you talk a little bit about and tell our viewers, how did you first learn about biosurfaces electrospinning technology?
Well, it's funny because I was at a angels investors meeting, and I always look for early stage technologies. And while I was there, a certain Matt Fainoff gave a talk on biosurfaces and the vascular grafts that they were making. And of course, we don't have a franchise in cardiovascular metabolism. We just recently gave that up. However, I got more and more involved when I saw how a vascular graft can be regenerated using this biomaterial where you have endothelial cells and then other cells eventually reforming almost like an artificial artery, right, based on materials alone. And when I saw that, I said, hey, well, if this is a new mechanism of action for a material, I wonder if this could be used for something in-house. And that led to discussions with clinicians here. And then a certain Dr. Dan Leffler suggested, hey, well, maybe we can use this as a fistula plug. And that's how I met Vitaly.
Yeah, I was gonna say that conversation led us, you and I to Dan to then Vitaly. So it was a nice progression of the discussion. And what made you decide to venture into the biomaterial space? This sort of bucks the status quo of just using a pharmacological intervention. To me, that was always, we've talked about this in terms of, to think about using that biomaterial as a pharmacological agent was really, I think, really innovative in Takeda. And so, what made you think about that in that sense?
Yeah, well, I mean, the thing is, I've always been in early discovery research, right? And to me, there are plenty, everybody else in the company can be looking for new drugs. It's okay. You don't need one more person who's done this for 20 years, right? If other people can take my place to do that, that's fine. And there's plenty of other people with that technical expertise. But I think what's more important is if you're going to innovate, you try to look beyond the box, right? I know right now we're in a Zoom meeting where there's a box around.
With all the boxes, right?
But you have to look beyond that, right? There's got to be something beyond the borders, right? And what I've learned, in my experience is that actually, the world, it's about the patient and it's about the therapy. And so to me, I proposed when I joined Takeda that I'm going to look at the therapy first. And if there's a technological intervention that uses drug to address the problem in the therapy, that's what I'm going to go for. I'm not going to go for something just because it's a new molecule, right? And so other people can do that. And so I think they could afford, one extra person to look outside the box in terms of this type of para-drug technology.
That's great. And so I'm going to bring Nate into the conversation here because, Nate, several years ago, I reached out to Nate when we were first starting our research and development program and we were building the team. And I went to Nate and said, we need your help. And so, Nate, you were brought on really specifically to work on this program. And you've been with the Fistula plug from its inception. And can you talk a little bit about the process that you and the team at Biosurfaces undertook with regards to the BioSpun J-plug prototype in terms of, the polymer choice, the rationale for design selection from a surgical standpoint, those kind of aspects that you could give some insight to our viewers?
Yeah, so after learning about some of the other options that were on the market and how most of those options were biodegradable, it seemed important to our team that using a non-biodegradable polymer might be able to give us an advantage in that area. Like Vincent has mentioned, the cellular ingrowth was pretty important for that. So the thought process of using a polyester would give us our necessary strength and structure for the device. And we've used this a little bit before in other applications. We've also used like polyurethane or PU, through our past experience, and that has been a little more stretchy, which can be helpful. But there's also some limitations to using polyurethane that we kind of stayed away from for this particular device. The design has been like a uniform material that can, be easily altered, so that a surgeon can change it up to fit in the tracks, because there's all different sizes. I'm sure Vitaly would be able to agree with that. So, the material has held up well throughout different types of testing, the tensile strength, suture retention, and we know that it can stay together when being implanted. So, another big aspect to the BioSpun J plug is the tissue ingrowth we've seen over the course of that testing. The material has shown a beneficial cellular ingrowth with non-surface modified materials, and has even better ingrowth when those materials are modified.
That's great. And, as I'm thinking about and looking at our screen here with our boxes, as Vincent pointed out, I look at, our goal has always been to, develop, a device that eventually the surgeon will want to implant, and having, Vitaly and Mauricio here, who have both been instrumental in implanting this, it leads me to think of how much we worked, with the surgeons on this, because ultimately, we want something that's they're going to use for their patients and that will help their patients. And I think having, the surgeons heavily involved in this part of the process was really so important, I think, in the development process, in any development process, and especially in this area as well. Can you, Nate, talk a little bit, we call this a J plug. Is there any significance to the name J plug?
Yeah, so I mean, there's some other products that are out there that may look similar to this, but we call it the J plug because the J stands for jellyfish, and it's kind of due to the round head and the tentacles or arms that come down off the product itself. So I guess the J plug, short, we call it here, like the jelly, is kind of what it looks like, and that's where we came up with that name for it.
Yeah, and I think, also just, thinking about that, as I'm thinking about the process that we've undergone to develop this, I think of the great feedback too from Vitaly and Mauricio as we were developing this. I remember Vitaly talking about the head of the jellyfish, and when we first developed it was too big. We needed to go smaller. We needed to do a couple of adjustments, and I think, so it really has been a real iterative process for us. Can you talk a little bit about the positive attributes you've seen with the device during the development process that you've undertaken?
Yeah, so over the course of the device, the strength of the material has increased as different iterations have been implemented throughout it. So not only is the strength been held up, but as I mentioned earlier, the reaction to cells has been great to see. The non-surface modified materials have shown cells to grow in within a few weeks. In even shorter time, the surface modified materials have shown cells to grow in a faster rate. And with even a little more ingrowth. So, the surface modification that we can do with these plugs is really important. It may have a little bit of an impact on the strength overall, but it really has been pleasant to see just the different types of things we can do with these plugs themselves and how they can be positively changed in different areas.
And what do we've had discussions within the group about, obviously we're in the very early stages of development of this plug. So we're at the very beginning. So for our webcast, folks, you're not going to see this in your stores, or you're not going to see this in patients in a year or 2 years. This is a time process that's going to take to develop this. And we're excited about that, undertaking that process. But, we always are thinking, similar to what Vincent talked about innovation and iteration. What's the next iterations you think that will occur with this type of device?
Right. Yeah. I mean, one of the first iterations was shrinking that head size, and we found a way to get the head size of the jelly or the J plug to be a lot smaller. So over the course of it, there's been some different iterations to get more strength, more surface modification to areas. One of the main iterations that we're focusing on right now are getting the arms to be cut more uniformly. Even trying right now, there's 4 arms that are cut by freehand, usually by myself or someone else. But so it's actually really difficult at times because it's hard to keep a straight line when all you're using is a razor blade or a pair of scissors or something just to cut that. So, one thing that we've been working on right there is trying to find a way to develop something to make that cutting process easier. that kind of is the next step in our line of trying to figure out how to make the product overall better. And getting consistency on the end is very important for us, which we've been lacking for quite a bit of time, but progress is being made, which is good to see.
Yeah, we're excited about, I think that, we're at the point that what you're talking about is switching it over from a research prototype over into manufacturing. And all of those processes need to be very controlled and very consistent each time. And we're already now heading into that aspect, which we're excited about. It's a complete change out of the research mode. I also think about areas that, like looking at the screen, I think of with Mauricio talking about, making the tentacles a little thinner. So actually adding some more tentacles. And that's one of the things that we're looking at in transitioning it from research to manufacturing. And I also think about what Vitaly talked about in terms of possibly adding in antimicrobials that he normally provides his patients, in terms of preventing this bacterial infection of the tract and of the device. So we have a lot of potential, already iterations, and I think that's the beauty of doing research and then transitioning it. So our first transition will be a non-drug loaded, fistula, J-plug over, through, the commercialization process. And for those of you who don't understand the commercialization aspect, this means going through the FDA approval process. So there'll be specific tests at the benchtop, as well as in preclinical, as well as potentially looking at it from a patient perspective, those aspects that the J-plug will go through, as every medical device does go through. And so that's our first iteration of this device. But these other things behind this are really exciting to us. And so thanks, Nate, for that, great insight in terms of how what the development process is. And another part of that process has been working with Mauricio. And Mauricio has really been involved heavily with the development of a preclinical model over a number of years, and especially for this specific device. And so, Mauricio, let me ask you, why is it the development of a preclinical model to test devices such as the J-plug important to develop?
It's actually critical, right? The model has to take into consideration several aspects. So most of these models have to meet very specific criteria. So in vivo studies will involve actually animals. They have to be in compliance with PHS guidelines, which are the ethical conduct for the care and use of non-human animals in research. So the model then has to mimic or duplicate the medical condition that afflicts patients. So for that purpose, for the predicate or the new device to be treated or tested. So in this particular instance, the use of the fistula plug is what's being proposed. So the protocol has to be approved by a committee at the institution before you can actually do anything. And these has to do recreate an anal fistula to then sort of like test the plug. So that's why, this part of the study is quite critical.
And I think these preclinical models are important. I think that, as I think of us as a research facility, there's only so many things that we can reproduce in a lab setting, right? You have, an animal provides you a very similar response to what a human would be in terms of having an immune response, having, having the same type, as you mentioned, the same type of disease state, if you will, or in this case, the same type of tract formation. Having these type of models is really critical. And so, what challenges did you face? And I know this answer from the inside because all the people on this screen have had these conversations of developing or trying to figure out what's the best preclinical model. So, what challenges did you face when we were developing these models? Because we've gone from a small preclinical model to a large preclinical model, and what challenges did you face in developing these type of models?
Well, like I mentioned earlier, most of these studies eventually will end up sort of like being in an animal model. So even though you take all the necessary steps to ensure that the animals are going to be well cared for and they're going to be comfortable, you cannot predict their ultimate behavior. So you can tell a patient not to touch, not to scratch, not to sort of like do anything as it pertains to sort of like, the plug healing. but the animal will do whatever the animal wants to do. So you sometimes have to be very creative in preventing the animal from, trying to dislodge the plug that has been implanted, particularly in the healing stage, between day 7, day 15. That's when, you have to really keep an eye on them, try to make sure that the plug stays in. For us, it's been very important to sort of like look at healing at different time points, and that being, 15 days and, 30 days. So you really want to make sure that the plug does stay in, because you really want to take a look at how the scaffolding or how the plug itself is actually really allowing or fostering, growth and, cellular growth and healing in the end.
Yeah, and I think you also bring up a good point in terms, and I think to stress this to our webcast viewers, is that the humane treatment of these animals is extremely important to all of us on the screen, and we understand that these preclinical models are critical in providing answers for patients. Could be, my parents or someone's grandparents or someone's parents who have to use this device. And so us understanding how this device is going to handle prior to getting into a clinical situation that Vitaly will be putting these into a patient is really, really vital. And I think that, I can't stress that enough, that I know there are groups of people who think that everything could be done at a test tube. And in reality, these type of devices, especially as medical devices, vascular grafts, heart devices, and these fistula plugs, there is such a complexity to all of the interactions that these devices undergo that we cannot duplicate that, in a benchtop setting. And we'd like to, ideally, we all would, but the reality is it doesn't exist for certain things. I think there are certain things you can, but for this you cannot. And so I want to I just wanted to just point that out. But let's talk a little bit about what you liked about the J plug in terms of the surgical implantation. Both you and Vitaly been heavily involved in implanting this device. Can you talk about it in terms of handling, adaptability, and suturability, which is important for these types of devices?
Sure. So first, we have to create, the anal fistula. So basically, we place a silicone round drain to create the fistula. We let it mature for 30 days, and then we go back in, and that's when we place the plug, your prototype. And it's actually a strong material that allows us to be able to drag the plug through the fistula that has been created once the setting is removed. And we do so, and the integrity of the plug itself remains intact, which is actually very important. Also, for us, it's important to be able to trim it or adjust it according to the size of the fistula. So it makes it easy for the graft to function. You were talking about earlier about the design of the jellyfish and having a head that it's too big. Well, sometimes we have to trim it so that it can be incorporated into the fistula, especially on the fixation aspect of it. The other thing about the plug is that we can actually suture it without sort of like the material tearing or breaking apart, which is actually also another thing that it's quite important. So overall, the material itself, even though it's sturdy and resistant to be able to suture it, it's also pliable in its design, and it allows for us to be able to trim it fairly easy. So I think these are things that we have to take into consideration. You don't want to create a plug or a prototype that in the end is going to be too hard and not comfortable enough for the patient, because they will eventually sort of like not like that, and the most likelihood is that you're going to have to have to go back clinically and take them out if this is going to be uncomfortable to the patient. So it's strong, and at the same time it's flexible.
I can speak for Nate when, I can say when Nate and I were in the OR with both you and Vitaly, and how much we learned, again, as the researchers, our job is to develop, the initial prototype, and then to learn from the end users who are going to apply it. And I think of how many things we learned on the first several surgeries in terms of, as you just mentioned, Mauricio, being able to adjust the head, I remember that was one of the first comments that both you and Vitali mentioned, and how you put it in and all the different things that'll led us to make a next version of this, with maybe a different head size and different tentacle lengths for our jelly design, our J-plug design. What did you see histologically? So histologically, for folks who are on the webcast, is if you look at these materials close up on the cellular level, really getting small, like what did you see when you took these devices out as compared to the current devices that are using that made you optimistic that this is a potentially good approach?
Well, before the histological, just the description macroscopically, what we saw on grafts or on plugs that were implanted at either 15 days or 30 days was actually quite interesting, because you were able to sort of like see almost complete healing on the 15-day plug on the anal canal by the mucosa. So that's actually quite important and significant. On the 30-day one, we saw full coverage of it. So I think you even have a picture of it. I think that we could show it, because I think it was very impressive to see the incorporation of the graft of the plug material into it and being completely healed on the luminal aspect on the anal canal. And then on the other end, on the skin where it was trimmed, I mean, we saw no discharge or no, evidence of infection, which is also, quite important. So that would be the macroscopically. This is when we harvested the material, and then we, fixed it and submitted to the core for processing for histology, what you were asking. So the histology was actually quite interesting as well. I mean, we saw that it had a moderate infiltrate of mesenchymal cells and dense collagen, with also sort of like red cells and mixed inflammatory cells. The penetration, the cellular penetration of the material varied. It was more robust and pronounced on the 30-day implants, as you would expect. I mean, the 15-day was, minimal, or there was infiltration. So for us, I mean, I think it's going to be very interesting going forward, is to allowing the plug to remain for a little longer than the 30 days and see what the full healing of it will be. And I think another change to the prototype, and this goes to what Nate was actually describing earlier, on the jellyfish being or having 4 different strands, or maybe even 8. If we could have the same material, but instead of having the same width of each individual strand, if you were to have sort of like more thinner strands, you would probably allow more of this healing, to take place. And particularly of importance, I think it was the finding that there were endothelial cells forming a preliminary sort of like a vascular network, which that is really, really important when you're healing and when you're incorporating a foreign body or a predicate, so that you can then really promote healing, overall healing. So I think that that's what's really interesting about the device. So overall, it was, and it was not me who, sort of like looked at the slides. We have a pathologist who you mentioned earlier from Takeda to take a look at them. And that was their interpretation, that the healing that's taking place is actually important and interesting.
That's excellent. That's a really great summary for our webcast viewers. And I do think ultimately at the end of the day with this, which we all agree with, and also what Vitali has pointed out in the past, is that ultimately we're going to need to see what these do in a patient. I think we will get, we will provide a, if all goes well, a very safe device to use. And then the question will be, as we transition from the preclinical to clinical assessment, does those types of descriptions that you're providing, does that translate over to the patient? And we're hopeful that if we do, the right studies, that we will see that, that translation over to the patient. And so, just before we close, I'd like to sort of ask each of the panelists their personal thoughts about the BioSpun J-plug in terms of what is appealing to them in their respective space, whether it be from the surgeon or from the research side of things. And also, what do you hope would happen with the J plug for it to be successful? So we'll start with Vitaly on that question.
I think there is definitely a void in therapeutic options for some of these patients. We have straightforward, easy procedures for folks who have a superficial and easy fistulas. But anything beyond that, we do have procedures that do work for a number of folks, but again, they're oftentimes very involved and uncomfortable and the success rate is not as high as we want it to be. So if we have a device that can either outperform those procedures, or even if it can show, I think in my mind, similar success rate, but with less involvement, with less interruptions to patients' lives, with less pain, shorter recovery, that would be a very big plus. In addition, there are definitely fistulas that are much more complex and require kind of multiple steps, where even things like lifts and advancement flaps don't work routinely. So that can be a possibility. And this is even before potential to impregnate that material with something else, with either antibiotics or some sort of other treating or an antibacterial or growth promoting solutions that can make this process easier. So, and as Mauricio was saying, I think they are easy to work with, which is important, because the harder it is from a surgical standpoint to do a procedure, the more frustrating it's going to be, but it's also more likely it is to fail. So it's easy to work with. It's kind of easy to implant. It seems to be very pliable, and so hopefully not causing a lot of discomfort. So I think there's definitely a void that I'm hoping we can fill with this.
That's great. And Vincent, your thoughts?
Yeah, so I think that this is our initial foray into biomaterials as a therapeutic modality, and I'm just really optimistic on where this can lead us. Because if this shows to have success in the clinic in the future, of course, right, it opens the door to possibilities for other types of fistulizing diseases and other indications, surgical indications where complex healing has to take place, and you do need some kind of substrate for a durable, I guess, basement membrane type of situation where you can have a better recovery of the wound site. So I have a lot of expectations that this will go far, and I think one of the things that people also need to understand is that there aren't many, if I would say maybe none, there are no electrosplin materials out there that's approved by the FDA to do this type of work, right? So even getting this through the FDA to allow it to expand to other indications, I think is just very exciting.
That's a great point. Mauricio, your thoughts?
Yes, I mean, I agree with, Vitaly and Vincent. I think it's very exciting that you have, come up with this new material, not new material in itself, but I mean, just the way that you've been able to electrospin this material into nanofibers seems to provide a scaffold for, vascular as well as cellular penetration. It gives or provides a completely different kind of healing from other biomaterials that we've seen in the past. And I think this is a golden opportunity now that you are creating the prototype to always keep in mind when you're doing this and when we're testing it, in vivo in these animal models, that you really want to think ahead in terms of what it will be for the patient to have something like this. I mean, if you start thinking, is this something that will be practical, off the shelf, ready to implant, like Vitaly was saying, easy access, and will give you ultimately the healing that you're looking for, that's where you have a successful product. The more complex that you make your product, or the more steps that it involves in being able to implement it, it's less likely that you're going to be able to, take it, all the way clinically. So simplicity sometimes is more, and being practical, and also thinking about, the patient itself. It's key.
That's a great point. And Nate, I'm going to change your question up a little bit, which is really going to stump you. But
Okay,
what would it mean to you, because you're one of the people that first started on this program and have been really developing it. What would it mean to you to see this device go forward and to help out a patient, one of Vitali's patients, for example, down the road? Like what would that mean to you in your thoughts of moving this forward?
Yeah, I mean, absolutely. I mean, going through school, I've always been intrigued by science, and in this area of just trying to help better people's lives. I think that's one reason why too I like this company and work here. So for me to be able to help push a product forward like this, from the research side, it would mean a lot to me that I could try and impact someone's life, make their life a little bit better, even from a minimal aspect side, from just helping to create a device and learning from everyone in this panel and on the team and things like that, of just how to change these iterations, and how to make a good product that just overall benefits people. So to me, that would be just really uplifting and reinforcing that I can really try and help others in any way possible. So.
No, that's a great way to end it. I would say, that for Vincent and I mean, I'll speak for Vincent in this. I think when we first started this discussion, we could only dream to be where we're at this point. It's a very big undertaking, and it really relied on everybody on this screen to get to the point that we're at right now. And so, we've got, still, the road ahead, but it's encouraging that we have such great people who all think about how can we help the patient, how at the end of the day, can we help improve a patient's life. And I think, to me, that's why I'm in this business. I think, that to me means anything more than anything itself. So I think, and I can speak for this group, that I know that that's how we all think. And so I'd really like to thank all of the panelists today for taking time to speak to us about, the perianal fistula, affliction, and your thoughts on, on how to treat this type of tract. And so thank you again for taking the time. If you have any questions, our webcast viewers, or you want to make a suggestion about future webcast topics, please feel free to reach out to us at info@biosurfaces.us, or to learn more, visit our website at www.biosurfaces.us. And we'd love to hear from you, and we're very appreciative of this panel being able to meet up. So until next time, thank you so much, and have a great day.